Bonjour à tous, à toutes. Aujourd'hui, c'est une nouvelle expérience pour l'USIAS. Je suis content que vous êtes là alors qu'il y a la fête de l'UNISTRA. Peut-être il fait plus frais ici qu'à l'extérieur, c'est l'un des bénéfices. Voilà, alors nous avons pour la première fois des leçons inaugurales. On a créé 3 nouvelles chaires cette année qui sont des chaires temporaires de 2 ans. Et on nomme des chercheurs exceptionnels, assez chers, pour faire participer à la vie de l'Institut et connecter mieux avec tout le campus. Alors, il faut que j'explique. Moi, je suis le directeur de l'USIAS. L'USIAS, maintenant plus de 10 ans. Elle a été créée sur le modèle du Collège de France. Alors, il y a beaucoup d'instituts d'études avancées dans le monde. Tout le monde pense que c'est celui de Princeton qui est le plus ancien, parce qu'Einstein y était. Mais en fait, ce n'est pas Princeton. C'est le Collège de France à Paris, qui est à côté de la Sorbonne, qui a été créé par quel roi? François Ier, il y a 500 ans. Presque dans quelques années, ça sera 500 ans. Ils vont fêter leurs 500 ans. Et ça a été créé pour créer une structure autonome, autogérée par les professeurs, à côté de la Sorbonne, c'est le bâtiment à côté, simplement pour les soustraire à l'influence du clergé. Okay, ça, c'est l'origine du Collège de France. Alors, voilà, donc on a une structure assez spéciale aussi, qui suit le modèle du Collège de France. Et dans ce modèle, si on continue l'étape suivante, c'est que tout le monde donne une leçon inaugurale. On n'a pas fait jusqu'à maintenant, mais on devrait. Donc tous les professeurs qui sont nommés au Collège de France, comme Nalini Anantaraman, par exemple, qui est aussi professeur ici à l'université, elle a été nommée au Collège de France et elle a fait une leçon inaugurale que vous pouvez voir en ligne, qui est super. L'idée, c'est d'expliquer leur recherche en des termes très simples, accessibles à un public très large. Et donc ça, c'est l'idée ici aussi aujourd'hui, qu'on va tester ici pour la première fois. Alors on a 3 chaires, qui ont un pour les sciences de la vie, un pour les sciences SHS, et puis une chaire pour les sciences qu'on appelle les sciences naturelles, c'est-à-dire chimie, physique, etc. Alors les deux premières chaires, on a maintenant nommé tous les 3, mais pour cette année, il n'y en a que 2. C'est pour la chaire de sciences de la vie, c'est Brigitte Kieffer qui est sur cette chaire, et ensuite on aura Frédéric Collin sur la chaire SHS.
Voilà, merci Thomas. Et donc c'est pour moi un très grand plaisir et honneur de présenter une neuroscientifique de haut vol comme première titulaire de cette chaire USIAS Paul Ehrlich dans le domaine des sciences de la vie, donc la professeure Brigitte Kieffer. Mais je m'étais dit, si Thomas ne parle pas de Paul Ehrlich, j'en dirai peut-être 2 mots, parce que je dois dire d'abord que j'ignorais qu'il avait été étudiant, probablement, j'ai pas trouvé exactement combien de temps, mais probablement 2 ou 3 ans, à la faculté de médecine de Strasbourg dans les années 1872-1875. C'est quelqu'un qui, par la suite, bon, c'était un Allemand, donc a travaillé à Berlin avec Robert Koch, et puis il a installé son institut, qui est devenu l'Institut Paul Ehrlich, à Francfort. Et il a fait des travaux, des découvertes très importantes et très applicables aussi, en hématologie et histologie, avec les colorants qui permettent de différencier spécifiquement différentes cellules ou différentes bactéries, comme la coloration Gram. Donc, en bactériologie, avec des études sur la tuberculose, sur la diphtérie, et surtout développer le premier médicament vraiment actif pour la syphilis qui, à l'époque, tuait beaucoup de monde. Et puis, l'immunologie, et c'est considéré comme un des grands précurseurs de l'immunologie, le premier à avoir proposé qu'il y avait des interactions anticorps, antigènes, et il a eu pour ça le prix Nobel en 2008. Donc voilà. Et, pardon ? En 1908, pardon, 1908. Oui, la vie a existé avant l'an 2000. L'Institut Paul Ehrlich est maintenant devenu, dans les environs de Francfort, la banlieue de Francfort, l'agence fédérale allemande pour les vaccins et la biomédecine. Bon, passons à Brigitte Kieffer. Moi, j'ai entendu pour la première fois une présentation de Brigitte Kieffer, ça devait être en 1990, ou peut-être une année avant. Et c'était à la faculté de médecine, au LGME. Et Pierre Chambon lui avait demandé de présenter son projet. Elle venait d'être nommée, je crois, maître de conférence à l'école des ingénieurs en biotechnologie à Strasbourg. Et elle a présenté son projet de cloner, d'isoler la copie cDNA de l'ARN messager de récepteurs opioïdes. Et elle était très convaincante. Elle avait une approche très innovante, mais dont à l'époque je pensais que ça va être risqué. Eh bien, j'ai eu complètement faux, puisque grâce à son talent, elle a pu effectivement isoler un premier cDNA pour, je crois, le récepteur opioïde delta, elle en parlera, et ça a été une vraie bombe dans le domaine, parce que, en fait, c'était quelque chose qui était un but très poursuivi par des laboratoires académiques, notamment aux États-Unis, et par des laboratoires pharmaceutiques, puisque les opioïdes, la douleur, etc., l'addiction, ça paraissait extrêmement important, intéressant. Et ce qui était absolument étonnant aussi, c'est que ça n'aurait pas pu être prédit par les critères usuels où on juge, n'est-ce pas, ceux qui présentent un projet, etc. Puisque Brigitte a commencé ses études en chimie bioorganique et sa thèse en chimie bioorganique à la faculté de pharmacie à Strasbourg, et elle n'avait jamais rien publié sur les opioïdes et les récepteurs opioïdes. À la fin de sa thèse, elle avait quand même travaillé à générer des inhibiteurs suicides ou des sondes photoactivables, voilà, des sondes photoactivables pour les opioïdes, mais je ne suis pas sûr que ça avait été publié ou ça a été publié, ça n'a pas été publié. Donc vraiment, on aurait pu se dire, mais qu'est-ce que c'est cette personne qui veut faire ce que des tas de labos très puissants, très riches, essayent de faire et qui n'a jamais publié dans le domaine, jamais non plus dans le domaine de la génétique moléculaire. Et elle y est arrivée, et je pense qu'à ce titre, c'est vraiment une illustration très importante pour les jeunes et les moins jeunes chercheurs, qu'on n'est pas prédéterminé par ce qu'on a fait tout à fait au début. Et donc, elle a ensuite continué à développer effectivement ces recherches qu'elle va présenter, mais au cours du temps, en changeant de stratégie d'étude au fur et à mesure que les problèmes se posaient, toujours avec la même idée de poursuivre les mécanismes d'action et l'utilisation possible, par exemple en thérapeutique, et la compréhension de la physiologie de ces récepteurs, mais en passant, après le clonage, à créer les premiers modèles souris, ensuite créer aussi des modèles innovants qui permettaient de suivre le récepteur en lui attachant une queue fluorescente donc dans la souris, et puis plus récemment en utilisant les méthodologies d'IRM à très haute résolution dans la souris, etc. Donc des travaux très importants. Brigitte a rejoint l'IGBMC en 2001, et notre département, qui s'appelle maintenant Translational Medicine and Neurogenetics, dont elle est devenue directrice. Et je voudrais dire que vraiment, c'était un vrai rôle modèle, comme on disent les anglo-saxons. Alors, où est-ce que je dis ça? Pour montrer comment on peut prendre des risques, on peut changer de technologie, etc. Et puis, elle est devenue, pendant 3 ans, directrice de l'IGBMC, avant de rejoindre, comme professeure de psychiatrie et comme directrice scientifique, un grand institut de neurosciences, enfin, sur la santé mentale à l'université McGill, et de revenir, donc ça c'était en 2014, et fin 2019, elle est revenue à Strasbourg. Oserais-je dire qu'elle n'a peut-être pas été reçue avec les égards qui lui auraient été dus vu tout ce qu'elle a fait, mais bon. Juste pour signaler aussi évidemment que ces travaux brillants ont été couronnés de récompenses. Le prix Richard Lansbury en 2004, qui est décerné par l'Académie des sciences françaises et l'Académie des sciences US. Le grand prix La Monica de neurologie de l'Académie des sciences en 2012. Elle est lauréate européenne du prix lauréat l'UNESCO en 2014. Membre de l'Académie des sciences depuis 2013, de l'EMBO, l'European Molecular Biology, depuis 2009, et chevalier de la Légion d'honneur et officière de l'Ordre du Mérite. Et donc, je suis vraiment très heureux qu'on puisse, qu'elle soit accueillie à l'USIAS d'une part, et qu'on puisse l'écouter.
Alors, merci beaucoup Jean-Loup. C'était une introduction très sympathique. Donc, je vais passer en anglais parce que c'était ce qu'on avait décidé. So, I would like to thank the USIAS for offering me the possibility to be a recipient of this new chair. I think I made most of my career at the Unistra, and it is a great university, and it's a great honor for me to be part of this institute. This scientific journey will take you through 5 points. So, first point will be a little bit on the history of opium, morphine, and opiate drugs to set a historical context. Then we'll move to the neurobiology of opiate receptors, which are at the basis of our research since 30 years, which will naturally lead us to try and understand or answer the question whether we can kill pain without producing addiction by developing opioid drugs. And this will lead me to tell you a little bit about addiction. What is addiction? What happens in the brain? It is a huge field, so I will only provide you with a few concepts to finally conclude on the biology of psychiatric disorders and a little comment I'd like to do about that. So throughout this journey, I will take you across disciplines and also across scales, from the brain to the atom, or from the atom to the brain. Point 1. Maybe a good way to start this presentation is to say that opium has been used since thousands of years because it relieves pain and produces euphoria. And in fact, it seems that this extraordinary substance is able to treat any kind of pain, whether we talk about physical pain, mental pain, social pain. There's all kinds of definitions for pain. And here you have a little timeline on the way opium was used. And the first writings or report of opium use come up from, well, step back 3,300 years BC, started in Mesopotamia. The plant was called the plant of joy. And the reports of opium use then spread and went to Greece, Persia and India, about 300 years BC, to finally reach China at about the 4th century. And medical records in Europe, well, in 17th and 18th century, medical records of opium use were spread all over Europe, Arabic countries, India and China. Actually, in the 17th century or 18th century, the opium preparation was typically called laudanum. You may have heard about it. And it was broadly used in Occidental countries, both for medicinal use. It was used for adults, but even for babies. It was used to treat everything, to everyone. And also for recreational purpose. And a number of famous writers, like Quincy in UK, used to say that happiness could be transported in a little bottle. And at that time, it was not so much question about opium addiction. But then things changed. There's a problem? Okay. Things change, and opium and opioid use has been at the center of societal challenges since a number of years and until now. You may have heard, of course, of the Opium Wars, where British trading policy flooded China with opium, which led to destroy the economy. A fourth of the male population was addicted to opium at that time. And then later in time came heroin use and heroin prohibition, and I will tell you a little bit about this in a minute. Until now, more recently, where the overprescription of oxycodone, which is a weak morphine, I would say, and also sold under the name of OxyContin, was overprescribed in the United States, leading millions of people to become addicted to OxyContin, then to switch to heroin use or to fentanyl use and abuse and overdoses. And in the States, there's many curves like this. There's about 1 person dying every 8 minutes from opioid overdose. It's a huge problem. It has spread out in Canada, and there's some signs of coming to Europe, but not so much because the medicinal control is much stronger. But if you're interested by this question, I suggest that you read The Empire of Pain. This excellent book is very, very well documented and explains how the Sackler family, through Purdue Pharmaceutical, oversold and pushed the prescription of opiates and started this opiate crisis. And there's also, for those who like series in Netflix, Dope Sick, that will also tell you about the story. This is happening now. So how does this work? So still a little bit historical. The most active ingredient of opium was isolated by a German chemist, Serturner, in 1805. And this molecule was commercialized then by Merck and was used intravenously in the hospital in 1843 from on, because the hypodermic syringe was created. So from there, in the hospital, morphine was administered intravenously and has much more drastic effect than opium, because this is not the pure molecule, and is highly analgesic, so it kills the pain. It's the most efficient painkiller, but it's also highly addictive. So from there, chemists started to work very hard. Can we design a molecule that would be slightly different and would not be addictive? So the company Bayer decided to make a very simple chemical reaction. So for chemists here, you will see you can do this in a kitchen. You deacetylate the morphine molecule, and you create a molecule, a wonderful molecule that was sold to treat asthma, actually commercial by Bayer. And this is heroin. So heroin was on the market to treat all kinds of diseases. And it took about 20 years for the medical doctors to realize that heroin was even more addictive than morphine, because it crosses the blood-brain barrier faster and it reaches the receptors that I will talk about much faster. From there, chemists have been trying to synthesize thousands of molecules, and I think today in available chemical libraries, there's about 10,000 compounds that are available. Here, this is just to show you a number of molecules that probably you know or you heard about. I talked about morphine and oxycodone. Fentanyl, I told you about fentanyl. Fentanyl, so morphine and oxycodone are used to treat pain. Fentanyl is used by anesthesiologists to diminish the pain during surgery. So it's in the anesthetic cocktail. It's very easy to synthesize, and now it's being abused like heroin in the context of recreational use. You may have heard about loperamide to treat diarrhea. You find in pharmacy something called Imodium, which is an antidiuretic. It is an opioid drug, and it simply doesn't cross the blood-brain barrier. So it doesn't go into your brain, and it acts on the GI transit and blocks it. It's an opioid. Naloxone is what we call the antidote down there. It is an antagonist, and this is the molecule that wakes up people dying from overdose. It will block the system, it will take away the opioid, and you will rescue the people that are dying. Dying from overdose means that the opioid is, I'm very sorry, is decreasing. Well, you stop breathing basically, because opioids act on the respiration centers, and the respiration is decreased, and you simply stop breathing. With naloxone, you'll come back to life. Usually, heroin addicts hate this because they come back to life and they are under withdrawal, and it's a very, very aversive state. But that's another story. You will see on the right methadone and buprenorphine, which are molecules that are sort of weak opioids, which are used as substitutive treatments. You heard about methadone treatments to help heroin or opioid addicts to go back to a normal life. And just as an anecdotal story, etorphine is a very complex opioid that is too strong to be used in humans. It is used in a veterinarian field. And for example, if you see an elephant charging on you, you send a little bit of etorphine and the elephant will crash. So that's just a number of very classical opioids that you find today in our everyday life. Let's move to the receptors. So before I move to the receptors, I'd like to get around and make a little description of the brain, from the brain to the atom, because there's a number of notions we need to know. The brain may look like, well, you may think of the brain as a complex machine, something we don't know much about, or whatever. It seems a mystery. But if you look through imaging, and you probably have seen images like this, MRI images that look at structural aspects of the brain show you fibers. The brain is full of fibers. This is a fiber tracking image of the human brain. And if you look at this fiber a little bit closer, you zoom in, and you will see that each fiber is attached to a cell body that forms a cell. And this cell that has fibers is called a neuron. And neurons have been highlighted, have been evidenced. The beauty of the shape of neuronal cells has been shown first by the father of neuroanatomy, Caral, in 1888 with special staining methods. And we know now that these neurons are organized between each other and form networks. And there are about 86 billion of neurons in the human brain. And each neuron talks to about 1,000 to 10,000 other neurons. So just to provide you with an idea of the complexity of the brain. The challenge of the 21st century for neuroscientists is to decipher what we call the human connectome, that is to describe all neurons and all connections of the human brain. And this is being done throughout the world in several consortia on several aspects. Now, I told you that neurons communicate. They don't talk to each other directly. This is my very simplistic scheme of what a neuron can be. So you can see the neuron here with a cell body and processes. And you see this neuron. So if the upstream neuron is activated, it will send a chemical message in this space in between the two neurons. This chemical message will then bind to a receptor on the further neurons, and the binding of this chemical message to the receptor will push the neurons to either work, thank you, that helps, to either fire or not fire. And there's many thousands of receptors here on each neuron that receive or sense chemical messages all around. And how does this interaction between the chemical message and the receptor occur? This is the very simple, very old images of the key and the lock. Basically, they have complementary shapes, and the chemical message will actually interact. They will form a complex that will tell the neuron, you know, work or don't work, or be silent or be excited. And because of this complementary shape, it means that any drug or any molecules that looks like the neurotransmitter, the natural transmitter, will be also able to bind to this receptor and usurp or take the place of the natural transmitter and push the neuron to react. And this is exactly what opioid drugs do. So how does it work? Basically, we know that morphine is going to act on the brain and produce strong analgesic and euphoric effects. In 1973, 3 labs, so there was a race to try and understand this, and 3 labs showed at the same time, to a US lab and a Swedish lab, to show that actually a radiolabeled opioid drug, so when I talk about a drug, it means a molecule, right, that binds to a receptor. It doesn't mean drug like drogue in French. It's a molecule binding to a receptor. So a radiolabeled drug will basically bind to brain extract in a reversible and saturable manner. So this tells us that there must be a given finite number of receptors on neurons that bind to these opioids. So if there are receptors in the brain, well, probably there's a natural endogenous molecule that normally activates these receptors. And the search for what we call opioid peptides was launched. And 2 years later, Hughes and Kosterlitz isolated the first, what we call opioid peptide, and you maybe heard about this molecule, enkephalins. Maybe you know better endorphins. These are the molecules that neurons release to activate opioid receptors. So in a way, we have a neurotransmitter system with an endogenous activator, possibly exogenous usurpator, if I can say it this way, and a receptor. Now, what is the nature of the receptor? And this is where gradually I started my research project. It was almost impossible to isolate biochemically the receptors to be able to know about their structure. These molecules are too rare and too unstable. So biochemists have been trying for years and years to isolate these receptors. It didn't work. So we had to get around and finally try and isolate the gene that we produce, the messenger that would produce the molecule, the receptor. And this is what we did. So Jean-Loup told you a little bit about that. I came here as a maître de conférence. I decided I would clone an opioid receptor gene. Everyone was trying to do it. I had nothing to lose. And why not? And we succeeded in 2 years with Claire Gavirio-Ruff and Katia Befort. It was not even a team. Chambon had hosted us, so we were able to go around the institute and work as we wanted. And we isolated a gene encoding a receptor called the delta opioid receptor, which led to, as soon as you had a sequence, you could go on and clone homologous genes with similar sequences. So genes encoding a mu and a kappa receptor were cloned. So it took 20 years from the discovery of binding sites to actually the gene cloning, the molecular structure of the gene encoding the receptor. And it took another 20 years until enough of the receptor protein could be produced artificially to be able to crystallize the protein, go for X-ray crystallography, and know the receptor structure, atom by atom. Okay, so now we know the receptor structure. I will come to this. In parallel, just to mention, cloning the genes encoding the opioid peptides was much easier. It was done in the 80s, during the 80s. And these genes, in fact, encode for precursor proteins that will be cleaved in small peptides, and there's about 20 peptides that naturally binds to the opioid receptor. We know them. You maybe heard about beta-endorphin, enkephalins, and dynorphins. This is their sequence, this protein sequence, and they all share a common N-terminal sequence here, which binds into the receptor site. Before I move to the structure of these proteins, I'd like to say that opioids are not the only external molecules binding to receptors in the brain and taking the place of a natural system. Cannabinoids bind to cannabinoid receptors the same way. Cocaine binds to dopamine transporters and inhibits it to increase dopamine levels. Nicotine binds to nicotinic receptors. Alcohol binds to all sorts of things. It's a very dirty drug, and it really messes up the system, but I won't go into detail here. But what I want to say is that all these drugs are consumed recreationally because they produce euphoria, because these receptors are all present in a circuit of the brain, which is called the reward circuits. Reward like récompense, right? Reward circuit produce pleasurable feelings. Why is this reward circuit absolutely essential for survival? Because there's a number of stimuli that need to be rewarding so that the individual and the species can survive. Food is rewarding, you eat to eat, otherwise you die. Social interactions are usually, but not always, but this is a kind of side, rewarding. And I mean, the strength of social reward is extremely important, you know that. And sexual activity is rewarding. This is because we need to live, we need to reproduce, and we need to live as groups to survive as a species. So this is a natural system which exists in our brain, which facilitates those behavior to repeat and eat again and so on. And this is what all these drugs are jeopardizing in the brain. Now, each of these drugs that I call drug of abuse, because they are potentially abused, has also other effects. So they are different. You know, they don't all produce the same effect because they are also present on different other circuits of the brain and do different things. Okay, so I don't go into detail. So now I go back to the opioid receptor. The opioid receptor, that was a very simple drawing. This is the way it is classically represented. When you know the structure, you can see that the protein has 7 transmembrane domains. It goes through the membrane of the neuron, and to finally form this little basket, which has a binding pocket oriented towards the external of the cell to receive messages, chemical messages, and has an intracellular phase to interact with effectors. And here we know the structure, so that's the way it is classically represented, atom by atom of a mu, a delta, and a kappa receptors that are bound to an antagonist, so they are inactive. So they were first to be crystallized and solved. And in 2015, we finally got the crystal structure of the active mu opioid receptor, bound to an agonist, an activating molecule. How does it work then on the cell? Well, this is the receptor. Here is the synapse. This is inside the neuron. When the agonist or the drug binds to the receptor, it will stabilize the conformation in such a way that effectors inside the cell now bind and get activated. And this is just an example of a structure of the receptor-effector complex. And when an opioid drugs bind to an opioid receptor, what happens is that the receptor will inhibit the neuron. It will inhibit neuronal excitability of the neuron. It will inhibit neurotransmitter release. Basically, it will silence the neuron. That's how it works. The effectors underneath, well, inside the cells, there's not only 2. There's a bunch of them. And here I simply show you the high number of effectors that have been described within the cell as being activated or modified by the binding on of a ligand on a mu opioid receptor. It is pretty complex simply because there's many effectors in the cell, because the signaling pathway, what is activated is drug dependent, even through the same receptor, because the presence of these effectors depends on the cell, and because signaling can also occur within the cell, because when the receptor is activated, it internalizes and goes inside the cell. So there's many molecular events that happened. And what I tell you here about opioid receptors is actually true for all these, what we call G protein-coupled receptors. Opioid receptors, in fact, are only a subfamily of a big receptor family, which counts about 400 receptors that we have in our body that respond to external hormone, neurotransmitter, or other molecules. And you need to know that in pharmacology, these mechanisms are extremely important. Half of the drugs you have in pharmacy to treat your diseases are actually activating or blocking this type of receptors. So it's a very important mechanism. So our opioid receptors are part of this family. Their conformation looks like this. Their specificity is that they bind opioid peptides. So now the question of can we kill pain without addiction? So now that we know what the receptor looks like, we need to move to the next step. We have 3 receptors. They have similar sequences, mu, delta, kappa. We know that in the brain, so this is one image, there's thousands of images where you can see that the distribution in the brain and in the spinal cord, actually throughout the entire nervous system, is different. Mu, delta, and kappa are not expressed on the same neurons. And we know from the pharmacology that they fulfill many roles. They are very important to reduce pain, all 3 receptors. They play on what we call our hedonic tone, where we feel euphoric or dysphoric. They play on our mood. They play on stress. The system is activated under stress. It's important that your opioid system comes up when you are in a very stressful situation so that you don't feel the pain and you can cope with the situation. And many others. I don't go into details. That are the main. So then the hope was we have 3 receptors, so maybe there's one receptor for pain and one receptor for addiction, in which case it would be very simple. We would design a drug that would activate one receptor for pain and avoid the other receptor. Well, no. Things are not as simple. So we went to the mouse. Why do we use a mouse model in neuroscience? Well, you can see that the brain is very small. It's down here. This is a human brain. But there's a high genetic identity. And also, with the mouse, we can customize the genome at will. And this started in the early 90s. And the lab of Pierre Chambon, for example, was one of the pioneers in these techniques, homologous recombination. So we could modify genes in the mouse. And also, they are good enough to behave, and we can study their behavior in a number of tests. So we can go from the gene activity to a behavioral response, and I will give you some examples. So what did we do? In a normal mouse, you can study if morphine reduces pain with very simple tests. You put the tail of the mouse in hot water, you see if the mouse will withdraw the tail. You can do this with your finger, with hot water. You withdraw your finger. It's a very simple test, but under morphine, the reflex is slower and slower because you don't feel the pain. So you can measure the analgesic efficacy of a drug with this test. Second test, you can measure if morphine is rewarding because you can administer morphine to the mouse, for example, to the animal, in a specific compartment, condition the animal to experience a drug effect with that context, then inject, for example, saline control in another compartment, and then a few days later, ask the animal, which compartment do you prefer? And with the morphine experience, the animal will directly go in the morphine compartment. Or you can offer the animal to self-administer to work to get morphine injections, and they do it. Okay? We can also study morphine dependence. We can study the constipatory effect of morphine in the animal. We can study the respiratory depression. We can see if the animal stops breathing. And we can study immunosuppression, because that's another effect of opioid drugs. Now, if we manipulate the mouse, we target the gene expressing the mu opioid receptor. The animal has no more gene, no more receptor protein. None of this answer can be measured. Morphine is losing all its effect. In other words, all morphine effects are gone. This means that a single receptor protein, a single trigger in the nervous system, is responsible for both the therapeutic effect and also the undesired effect. So this is a true problem, and you can actually check. We prepared also mice that have no delta receptor or no kappa receptor, and these mice react to morphine perfectly normally. So the mu receptor is indeed the receptor for opioid drugs that we know, that are used in the clinic and that are abused. So how can we deal with this? Now we have a single receptor to target. Okay, we will necessarily produce addiction and analgesia. Well, second question is, where is the receptor in the brain? In which neurons? And we were able to develop an interesting animal, and I want here to thank very much the mouse clinic in Strasbourg, and in particular Marie-Christine Birling, because it took us 10 years to produce this mouse successfully. And this mouse allows us to visualize all the neurons. Okay, this is not going to work, too bad. Okay, I had a movie, but you won't see it. You can see all neurons that actually produce a mu receptor, respond to morphine. And you will see that there are many, many cells throughout the brain that actually respond to morphine. Okay. We could even refine the approach, and we created another cell, another mouse, sorry, that will now express not a fluorescent or produce fluorescent cells, but a fluorescent receptor on the cell. So now we can see the receptor, and we can scrutinize all brain circuits, and we can see that this receptor is expressed all along reward pathways that I mentioned earlier. For example, in those brain areas, what you see here is the receptor. So this is happening here in the brain. It's actually in the midbrain. And if we look at pain pathways, which are going from the periphery to the spinal cord, to the cortex, and back down to the motor neurons, well, the receptor is also expressed all along this pathway. So I'm not going to talk much about pain here or pain control, because I had to make a choice. But if you want to know more about pain pathway, there's an excellent Pain Day tomorrow at Unistra by Yalcin, Ipek Yalcin, and also you can go and read this review by Greg Chaire, which explains you everything. Okay, I will more focus on addiction. So in a way, this means that if we develop a new drug like morphine, we will necessarily produce reward and reduce the pain. So is there a way out of this? Is it totally desperate? Well, people have been extremely creative. And I will just tell you about one way that may offer a solution. Not totally proved yet, but it was shown that I showed you this mu receptor binding to different effectors. One is called beta-arrestin, the other is called GOGI. Two different proteins, two different pathways, and two different messages within the cell. Well, it was shown that if you delete this one in a mouse, you produce less secondary side effects. So maybe the idea was to develop drugs that would bind to the receptor, but rather activate the blue pathway here. And this was done. This was done through the docking of millions of molecules virtually on the receptor structure, or through classical screening using recombinant receptors. And molecules were produced that actually bind to the receptor, produce analgesia through the blue pathway. But whether there's less side effect is not as clear. One of these drugs is in clinical trials. The analgesic efficacy is kind of not conclusive. So we're not out yet, and there's a debate about the fact that maybe simply less efficient drug would produce less side effects, which kind of makes sense. This is a joke. So one way is to use low efficacious opioids under strict medical control. or maybe to target other receptors and avoid the mu-opioid receptor. There's a huge research field about other targets to treat pain, but in fact, none of them is as strong as the morphine effect to kill pain. So that's where we stand today in the field. So now I will move from, well, a little bit to addiction. Okay. Before I move to addiction, I'd like to say that, as I told you before, producing reward is natural. And in fact, what we found is that the mu-opioid receptor, that is the main key for morphine, is not only the main key for morphine, it's the main key for reward. Why? Because if we delete this mu receptor in the mouse, as I said before, not only do we totally ablate or delete morphine reward, but the mice will not show any appetitive or will not show, well, alcohol will not be rewarding in these mice. Cannabis? Mm-mm. Nicotine? Neither. So it seems that this receptor is a convergent target, that all these drugs will activate at their own receptor, that will in turn trigger enkephalins in the brain, that will activate the mu receptor, and this one is a central piece for the rewarding effects or the recreationally appreciated effects of drugs of abuse. And if this is the case, we ask the question to know whether this receptor is also central for natural rewards. And we were indeed able to show that motivation to eat is decreased in these animals, and also that we were able to show a social interaction deficit. And this was a very, very nice study that was developed by Francesca D'Amato in Rome. We used to study maternal attachment between a mouse and pups. And what she showed is that pups that had no mu receptor since the very beginning, that were knocked out for the mu receptor gene, show much more reduced maternal attachment to their mother as a control pup. Meaning that, in fact, the maternal behavior is normally rewarding to the pup, and the pup will behave as to have the mother reproduce the behavior. And this is the way bonding takes place. In this case, maternal behavior is less rewarding to the pup, and the bonding does not take place as well. So it is a key molecule. And now, so that's it. Addiction. So now let's move from a normal situation to a pathological situation. What is addiction? For the clinician, okay, it is complicated. There are 11 criteria to diagnose addiction for an individual, and you have to fulfill 3 criteria among 11. In fact, I am not going to describe them all because it would take too much time. It is sufficient for you to know that at the moment, the consumption of the drug is done despite what we call negative consequences. That is, the person is losing his health, is losing contact with family and peers, is losing job, everything's gone. Then you have the diagnostic of being addicted. This applies not only to opioid, it applies to a number of illicit substances like psychostimulant, I mentioned cocaine or amphetamine, cannabis, licit substance, alcohol is one of the worst drug of abuse, tobacco also, and there's we call non-drug addiction like gambling, sex or food addiction. So, recreational drug use is not pathologic. I won't insist too much on this, but there are statistics showing that, in principle, recreational drug use can be done, and only a small proportion of people will actually enter into addiction. About 15%, depends on the population, depends on the individuals, of course, but not everyone will become addicted. But once your recreational consumption switched and becomes binge intoxication, or you consume, this is what we call binge intoxication, then you switch from recreational use to abuse. And this is a way psychobiologists have conceptualized what we call the vicious cycle of addiction, because it's easy to understand. Basically what happens is you go through your binge intoxication episode, after which the drug clears out of the body, and then you feel under intense withdrawal. You're lacking the drug, because now your receptors are empty. This state is extremely aversive. Then you go into what we call a craving phase, trying to search for the next drug consumption episode. And you go back there. And the more you do this, the more the euphoria is decreased and the withdrawal is increased. I kept the color code here to show which brain areas, and we know very well which brain areas are actually recruited and engaged in each step or stages of the vicious cycle. We know where the things happen. And basically, if we summarize what happens in your brain adapting to chronic drug exposure, repeated exposure, reward decreases, as I said, withdrawal and negative affect increase. The stress response is enhanced. Your responsibility to stress is enhanced. Motivation is totally biased towards searching for the drug. That's your main motivation. Automatic behaviors are enhanced, and self-control is weakened. And decision-making is very poor. And when one says that it is only a matter of will, this is totally wrong. The brain has changed. You're not able, actually, to make the good decision. And exiting this vicious cycle is indeed extremely difficult. And the true challenge for an addicted individual is to get out of this cycle and maintain what we call protracted abstinence. So stay abstinent without consuming the drug for months and months and months and years. And it is known that actually the more time passes, the more what we call negative affect is increasing. In fact, what we see in the clinic is that mood is getting lower and lower, irritability is increasing, emotional responses are enhanced. In fact, the abstinence state is a true problem. Even if physical withdrawal is gone, you still have this, what we call psychological withdrawal, that maintains, I would say forever, but almost. So this is very much studied. But the neurobiology of protracted abstinence, that is what happens in the brain after a long, long period of abstinence. You have not seen the drug since years. What happens? Why is it so hard not to relapse? So I'd like to mention here, because I would like to go to an experiment, right? Why not talking about an experiment? We have developed a model of what we call protracted abstinence to morphine. So our little mice have received morphine very heavily during a week, high doses of morphine. So they have become terribly dependent. And then they go back in their cage quietly. They grow physical withdrawal. We can see that. We can measure that. After a week, physical withdrawal is gone. And then we let them just go. After 4 weeks, which is a very long time for a mouse, it could be a month for a human, let's put it that way, what we see is that these animals gradually develop what we call a despair-like behavior. So this is well, I'm not going to go into detail into the test, but they stay more immobile. They don't fight anymore. They get sort of depressed-like. And what we find also is that their social interaction diminish. They are not interested in socially interact anymore. And this is not at the beginning of abstinence. This develops with time. So there's this incubation of what we call low mood. And we were able to show that this is also seen if we use heroin, another opioid. This is long-standing. It can go until 10 weeks. And in fact, we didn't look how far it will last. We know that serotonin is involved. And for those of you who don't know, serotonin is the neurotransmitter controlling mood. And we know that we can prevent the development of this behavior or misbehavior using Prozac, which is an inhibitor of serotonin uptake. it's the classical antidepressant. So there are mechanisms in there that are changed in the brain for a long, long time. So what is the dysfunction? What is the dysfunction? So here a little methodological tour on one quick slide. We can study how genes and molecules and receptors act on neurons and how these neurons will interact together and will control behavior in a different ways. We can work at microscale level. What I call microscale is we can manipulate the gene, the receptor, the cell. We can do this with all these fancy techniques in neuroscience, and we can tweak every little neuron in every place of the brain to try and understand. There's another way to do things, which is to zoom out and look at mesoscale level, which means look at how brain regions talk between each other. And this can be done by functional MRI. So it's not the same resolution at all, but it's a different view. It's a brain-wide view. And I have personally been very interested in having this view of the brain. And why is it important? It is important because it is non-invasive and it is translatable to human. How do you look at brain functioning in humans? The best way to look at brain functioning is what we call functional MRI. So you go in a scanner, and then you can look at brain at work, the brain at work, and you can, for example, relate genotype to phenotype, find biomarkers for diseases. You cannot study drug effects so much for ethical reasons. But in this case, in human, this is what we call correlative research, because you're going to connect or link, associate what you see on the image with what you know from the person's behavior. Now, why wouldn't we use the mouse? Knowing that with the mouse, we can do mechanistic research, because in the mouse, we can manipulate the gene and look at the consequence. So this is causal research, mechanistic research. But again, I'm coming back to this scale issue. This is a human brain, this is a mouse brain. And this means that for a little mouse brain, you need a huge scanner. And this is when I met Laura Arsan. She was working with one of the Pope of MRI in Freiburg. Is now retired. Laura is now the head of the IMIS team in Strasbourg. And she was at that time the only person I knew that was brave enough to put a mouse in a scanner and to develop high-resolution analytic methods to study the mouse brain, which is like this, okay? And then I went to meet with her. I told her, can you make a difference between a normal mouse and a mouse that has no more mu receptors? Is there any difference? And honestly, I didn't think we would see any difference. But we did. So I don't show you the details, but we published that in fact, in these animals that had never had a mu receptor in their life, what we call the reward and aversion connectome, so the circuits that process reward and aversion, are changed. So we went on, and this is now unpublished data that I'd like to share with you quickly, because I think this is also the purpose of this talk, is also sharing the excitement of science, right? So this is not published yet, but what we did is we prepared abstinent mice, exactly the same way I told you before. So these mice have not seen morphine since 4 weeks. And we put them in the scanner. We do a scan at rest, and then morphine comes in the body of these mice. And we keep on acquiring images. So we have these mice at rest and then under morphine. And after the imaging is recorded, then we verify that they indeed show the social interaction deficit and the despair-like behavior that we know. And why did I put all this? Because this is really a team effort. All images were acquired at the Douglas Research Center in Montreal, and all the analysis was done here at iCube with Laura Arsan and the platform there. What do you measure, in fact? What we measure are Ball fluctuations. So it's blood oxygenation level in the brain, and this is a proxy for neuronal activity. It's not neuronal activity, but it's like neuronal activity. And what you see in the brain is that there's a signal like this. And you can divide the brain in about 100,000 little volumes. The mouse brain, 100,000 volumes, we call voxels. And you can measure the blood fluctuation in each of these voxels. And that's what you're going to do, is try to correlate. See if some voxels or group of voxels are synchronized. And you say that if they are synchronized, they are functionally connected because they work together. So we have these mice that have control mice that have not seen morphine before, go in the scanner, baseline, morphine. And we have these mice that had the history of morphine exposure, then gone through abstinence, go in the scanner, and then baseline and morphine. We chose to look at 24 brain regions, which we know are involved in addiction, the one I mentioned before. And we looked at the correlation or the synchrony of the 276 potential connection between each other of this brain region. And what we found, so I don't show you the data here because it's complicated, but we looked at connectivity pattern, and we found that indeed there was a difference between abstinent and control mice. There was a difference, and their responsivity to morphine was different. And what I'd like to share with you is that we went a little bit further and looked at voxels that form networks in the brain. So these are brain regions that are synchronized in the brain, in the mouse, normally. And once we have identified, we had 19 networks, we decided to compare between the control mice and the abstinent mice. And we found two types of results. The first result is that if we only consider naive mice, we find that there's morphine entering into the system for the first time is changing several networks. I show you 3 networks here in the brain that are changed by morphine. One is the reward and memory network, the other is dopamine and serotonin systems, and the other are cortical networks. Now let's look at the abstinent animal that has not seen morphine since 4 weeks, totally abstinent. What do we find in these animals? Well, 2 of the deficiencies here, 2 of these networks, remain dysfunctional all this time. They don't get back to normal. So we have this morphine trace in the brain of the animals. And we also have another marker, which we do not see in control mice, which is a dysfunctional, what we call default mode network, which characterizing the resting state. So the animal is not under resting state anymore. Why is it important? Because I think not only have we shown that there's long-term changes, but we have been able to identify a long-lasting morphine trace in the brain. We have been able to identify a brain dysfunction. Well, in the future, we hope that we can translate some of this knowledge to human imaging, which is much more difficult for many reasons. Humans are very different from each other. They have different drug consumption history. And yeah, there's too much heterogeneity. So maybe we can find some traces or some markers that we could use perhaps to predict a treatment outcome and to personalize the treatment. So this is a kind of vœu pieux that we would have in the field. And to finish, I just like to make a little comment. Okay. I started at 2:15, huh? Okay. I hope, so almost last slide. I hope I convince you that we can learn a lot on the brain, and the opioid system is a kind of very nice illustration of how you can start from a plant and ultimately, gradually learn so much about, in this case, pain pathways or reward pathways or others, and about brain dysfunction. I'd like to mention that there's still a very big disconnect in the scientific community between basic neuroscience and psychiatric research, which is really far from it. Why is it so? Well, psychiatric illness or mental illness are scary for you and me, because you have a kind of dysfunctional behavior that we don't understand. And mental ill patients are usually stigmatized, less today, but have been very much stigmatized. The reason we don't understand, and it's so hard to address mental illness, Well, there's three main reasons. The symptoms are extremely difficult to describe. You go through questionnaires, right, with patients, and they are different from one patient to another, very different. There's no biological marker because you cannot punch a little piece of the brain to try and decide what's wrong, as you would do for a cancer tumor, for example. And this is the reason why psychiatric research has lagged behind cancer research by at least 20 years, if not more. And finally, there's only failed clinical trial in the pharmaceutical industry. Why? Because the brain is so complex, and it is very difficult to create cohort of patients that are homogeneous enough to have a clinical trial to be successful. But we could debate on that. But so all this is extremely difficult. So I hope in the future, I think what is important is that gradually neuroscience will feed more and more the mental illness area, and that will be able to bridge the two fields. And a lot of people are working on that. So to conclude, as far as our research is concerned, this was an incredible journey, 30 years, from receptor cloning to molecular neurobiology, to cellular biology and molecular pharmacology, understand how these receptors function in cells, then how these receptors function in vivo, what is their roles, and then we even went on discovering what is the role of receptor within specific brain circuits. I didn't touch on that, to finally go to maybe from circuit neuroscience to computational neuroscience. It is important that all the bridges are filled, and I want to thank many, many people. I want to thank mentors here, my two PhD supervisors. I indeed did a master degree in organic chemistry. Pierre Chambon, who hosted me and allowed me to do whatever I wanted in his institute. My international collaborators since 25 years. And all the teams I had at IGBMC, at the Douglas Research Center, and now the small team I created at the CRBS to hopefully give the hand to younger investigators. And also a special thanks to Claire Gaverio and Katia Befort, who started this adventure with me, and I thank you for your attention. Thank you. That was probably too long.
I'll take this one. Keep this or the other way around. Maybe we need for the audience if somebody. Oh, they have mics. OK. Alors, merci pour cette présentation remarquable. On peut revenir en français, je pense. Les gens peuvent poser des questions dans les deux langues. Et voilà. Est-ce que vous avez des questions? Des questions? Bon, moi, je vais. OK, il y a quelqu'un là-bas.
Vous avez utilisé le mot addiction et dépendance, et je me demandais quelle différence il y avait entre les deux, parce que je n'avais pas fait la distinction.
Vous pouvez mettre votre...
La dépendance, c'est un aspect de l'addiction. La dépendance, on utilise souvent les termes pour les opioïdes, tolérance et dépendance, qui sont des manifestations effectivement du problème. La dépendance, vous la voyez quand vous êtes en sevrage. C'est-à-dire quand il n'y a plus de drogue et que vous êtes en syndrome de sevrage. Là, on dit que vous êtes dépendant. Mais c'est juste un petit aspect dans le cycle dont je vous ai parlé. C'est un des aspects des dysfonctionnements.
Oui. Moi, j'ai... OK. Je sais pas ça, oui. Allez-y. Mettez le micro, s'il vous plaît. Il y a un petit bouton.
Oui. Je me pose la question de la fin de la conférence, qui est de savoir pourquoi la psychiatrie n'a pas avancé dans les neurosciences. Mais j'ai une réponse, c'est l'inconscient.
Oui, mais...
Oui, je voudrais quand même une réponse.
C'est quoi la question exactement? Pourrez-vous formuler votre question?
À partir du moment où tout est dans le cerveau,
oui,
dans les neurones, dans les messagers, dans toutes ces choses-là, qu'est-ce qu'on fait de l'inconscient?
Mais il est aussi dans les neurones et dans les messagers. Tout est dedans, inconscient et conscient. Les circuits, on connaît bien en recherche humaine. Bon, je ne suis pas spécialiste de recherche humaine, mais on connaît très bien le fait qu'il y ait des réseaux qui fonctionnent de manière consciente et il y a des réseaux qui fonctionnent de manière inconsciente.
Oui, mais c'est sans drogue, c'est hors drogue.
Oui.
Oui.
Drogue et inconscient?
Oui.
Oui, ben... Alors, il y a une remarque que j'ai entendue un psychiatre me dire un jour, qui m'a dit, c'est catastrophique si l'événement va directement dans le système limbique sans passer par le système cortical. Les drogues vont directement attaquer votre système limbique et ce qui se passe dans votre système limbique, il n'est pas conscient. Ce qui est conscient, c'est ce qui apparaît au niveau cortical. Donc les drogues, quand je vous montre les circuits de récompense, ce n'est pas des choses conscientes.
Oui, mais l'inconscient, c'est une histoire ontologique. Ça n'a rien à voir avec ce qui rentre juste dans le système limbique.
Absolument, mais là, on rentre dans la discussion entre neurobiologistes et des gens qui s'intéressent à la santé mentale. Et la question, c'est qu'on n'a pas du tout le même discours. Les mots ne veulent pas dire les mêmes choses et la complexité n'est pas la même. Et c'est pour ça que je milite pour que les deux disciplines se parlent.
Non, mais la question, c'est est-ce que vous guérirez? Parce que c'est ça le sujet, finalement.
Ok.
Bah, oui.
Est-ce qu'on guérit de l'addiction?
Non, est-ce qu'on guérira à partir des neurosciences?
Ben oui, moi j'y crois dur comme fer.
Pas moi.
Ben voilà, on n'est pas d'accord. On peut en discuter si vous voulez.
Jean-Louis.
Oui, je t'avais trouvé peut-être un peu négatif sur le fait qu'il n'y ait pas eu d'avance dans la santé mentale. Alors, évidemment, une fois qu'on a une certaine avance, on voudrait aller plus loin, et quand ça bloque plus loin, on trouve que ça ne marche pas. Mais quand même, il y a des médicaments qui aident certains, pas tous. On ne sait pas prédire toujours qu'est-ce qui va marcher chez un, combien de temps ça va marcher. Mais il y a quand même des médicaments qui ont été développés, des antipsychotiques, etc., qui posent des problèmes, qui ont etc. Mais enfin, dire comme ça qu'il n'y a pas de... C'est un peu dur, non?
Je suis d'accord avec toi. J'étais un peu noire et blanche là, j'aurais pas dû. C'est vrai qu'on a fait beaucoup de progrès dans les médicaments. On a amélioré peut-être les médicaments existants. Mais pour ce qui est de trouver des nouvelles cibles et des nouveaux mécanismes, là, les essais cliniques ont systématiquement loupé. Et la raison pour laquelle, par exemple, le médicament pour la schizophrénie de Roche, qui était allé en phase 3, ils étaient prêts à commercialiser le médicament et ça a raté. Et la raison pour laquelle ça a raté, c'est que quand on essaye le médicament en phase 3, on va rassembler des cohortes de malades et des cohortes de contrôle. Et les cohortes de malades, eh bien, c'est des diagnostiqués schizophrènes, on va dire, dans ce cas-là, avec peut-être des sous-classes de schizophrènes. Mais dans les faits, ils ont peut-être des maladies biologiques différentes, ils sont classés sous le même chapeau, et le médicament ne marche que peut-être sur 10% d'entre eux. Donc quand tu fais ton analyse clinique, c'est inconclusif, il n'y a pas de résultat significatif. Et c'est ce qui arrive encore et encore. Donc il faut pouvoir stratifier les patients sur une base biologique de façon à faire des essais cliniques sur des sous-groupes de patients qui auraient une chance de répondre. Donc,
mais ça arrive aussi dans d'autres médicaments, on en a plein. Sylvia le connaissait très bien. Ça, c'est un problème de beaucoup de drogues aujourd'hui. Elles marchent sur certaines cohortes ou certains profils, mais peut-être la génétique peut faire la différence là. Il y avait une question là-haut.
Yeah, so I think it touches just what you are talking about. So you mentioned that the cycle of addiction, some people, about 15% of the population, might get addicted and the other don't. So is this something that we know what is creating? Is just what you're saying, like genetics? Is this...
Yeah, well, everything is, genetic and environment, right? Is the combination, of course. So this combination makes that some people will consume recreationally but have a genetic background and an environment that make them less susceptible to fall into the intoxication stage. Others are more susceptible. It's the combination of both, but we have no description of which genes and which history well, for historical reason, we may, you know, there's environment that are more prone, where drugs are more available, where you may push the statistics a little bit. But, yeah. Did I answer your question? It's vague.
Yeah. Well, I guess it's a combination of both things. It's difficult to isolate one.
Yeah.
Ok, encore, il faut qu'on ait une petite pause café. Vous pouvez poser des questions. En dernière question, le monsieur, là.
Merci, j'avais une question sur la partie imagerie. Donc, tu disais en fait que chez la souris, c'est du causal, parce qu'on peut connecter la manipulation à la sortie. Par contre, en fait, le lien entre la manipulation génétique spécifique qui a été faite et la sortie au niveau des changements de l'imagerie reste encore corrélationnel. Est-ce qu'on peut, on fait cette manipulation, on observe ce changement, mais est-ce qu'on peut renforcer le lien en essayant de modéliser, par exemple, l'impact que le changement génétique, le changement sur les canaux aurait au niveau de la dynamique? On connaît l'anatomie après tout, en fait.
Oui.
Est-ce qu'on peut aller au-delà de la corrélation avec l'effet d'imagerie et expliquer l'effet d'imagerie à partir du changement au niveau des canaux?
Oui, alors déjà, pour moi, je trouve que ce n'est pas une corrélation, parce qu'on a réalisé une manipulation qui a permis de produire des...
Qui corrèle avec un effet au niveau de l'imagerie.
Oui, alors maintenant, si effectivement tu veux corréler l'absence... Ouais, oui, on pourrait aller dans l'autre sens. Je sais pas, il faut qu'on en discute. Okay. J'ai du mal à répondre précisément à cette question.
Des questions fascinantes, la recherche de Brigitte. Donc, merci encore à Brigitte pour cette belle présentation. Et on a une pause café et ensuite on passe à l'Égypte.